We're recording now. Okay, we're going to at least get through cell division today. We've got around 25 minutes left. I think that's a pretty manageable goal. And again, so this is section 2.31 in that link that I posted. So tomorrow by midnight, you've got to answer the review questions for this. Does that sound fair? And then we're probably not going to have class tomorrow. So, but. Okay, so cell division is the process of cells dividing. There's an old joke in biology that it says cells are the only thing that can multiply and divide at the same time. Right? The joke is because that is how cells multiply, by dividing. They grow and divide, they grow and divide. They grow and divide indefinitely. So in this section, we're going to talk about what is cell division, and then the processes in prokaryotes and eukaryotes. What's the difference between those two things?
Prokaryote has a cell-bound membrane.
Prokaryote.
What did you say?
Cell-bound membrane.
What did you say before that?
Prokaryote.
No.
No, it's
Really?
eukaryote.
Yeah, eukaryotic organisms, eukaryotic cells, have a membrane-bound organelles and a nucleus. Okay? Eukaryotic cells, membrane-bound organelles, and nucleus. Prokaryotic cells have neither one of those things. Okay, prokaryotic cells are more simple. Prokaryotic cells are bacteria. Eukaryotic cells are things like animals, plants, fungi, etc. So if you look at this picture, you can actually see this is cell division occurring right here. A specific type of cell division that happens in eukaryotes called mitosis. Alright, cell division is a process in which one cell, called a parent cell, divides to form two new cells called daughter cells. Parent cell is the starting cell that forms the two daughter cells, and the daughter cells are the new cells that result in cell divisions, i.e. the divisions from the parent cell. This is key for growth and development in organisms from a single cell to trillions of cells. So anything that grows does this. Alright? Anything that grows, even bacteria go through this. Again, this is just an example of you've got the parent cell that can split into two daughter cells. As you remember, prokaryotic cells are usually a bit more simpler. They have one chromosome. It's usually in a ring. They have no nucleus. Signified up here is this visual. Eukaryotic cells, these are more complex. They've got multiple chromosomes in a nucleus along with other organelles. Just as kind of a recap, we'll see if you know this. What organelle is this?
Mitochondria.
Mitochondria. What's this one? This one's going to be the Golgi. What's this one? Speedy R. Ruffy R. What's this thing called? Nucleus. What's this dark matter inside the nucleus? Nucleolus. What are these little dots? Ribosomes. Very good. What is this?
Vesicle.
Vesicle. Very good.
You said what?
Come on in. Alright, chromosome is a coiled structure made of DNA and protein. Where's that going to be at on this one? Where's the chromosomes?
In the middle of it.
Yeah, it's all this stuff. Where's the chromosomes for this one?
It's in the middle.
Good. Okay. You can see those chromosomes are contained in the nucleus of eukaryotic cells. Alright, in prokaryotes, the type of cell division is called binary fission. It's a method of asexual reproduction that occurs in prokaryotic cells in which a parent cell divides into two identical daughter cells. So this happens in prokaryotes, so think bacteria. Again, these are identical daughter cells. Genetically identical meaning they'll be same. Very good. This is going to have three different steps. DNA replication, where you double the DNA. Chromosome segregations, where you have these separation. Say what?
So segregate the chromosome.
Yeah, separation of chromosomes to opposite ends of the cell. And then you have separation, which is the formation of two daughter cells with their own chromosomes. Again. Any questions on that? Easy? Easy peasy, lemon squeezy? Alright, visually it's going to look like this. You have the DNA replication where it doubles. You have the chromosome segregation where it splits those chromosomes into opposite parts of the cell. And then they finally separate. It's also going to look a little bit like this if this is a little bit better visual for you. Alright, now just a question. Do you want the on-level notes for this to fill out as we go along? Okay. Alright.
Quick question.
Yes, sir.
What's the purple stuff around it?
Which one?
Like the purple stuff around it.
Cell membrane?
Okay. Why is it like
It's separate.
Yeah, why is it getting bigger though on that one?
I don't know, because the graphic artist wasn't very good, maybe.
Splits.
Yeah. It does split, so you do have to make it bigger so you can split and have enough for these two new cells. But I'm not sure if that's what they were trying to do, or if it just kind of happened because they're not very good at drawing. All right, in cell division eukaryotes, what is this process? Starts with an M. Mitosis. Okay, this is going to be more complex because there's a lot more stuff going on. Remember, prokaryotes don't have organelles. Eukaryotes do, and so there's a lot more moving pieces, and so it's a bit more complex. Steps here are going to be two: mitosis, okay, and then cytokinesis. Mitosis is the division of the nucleus and the organelles into two identical nuclei before the cell divides. When we get to genetics, there's two phrases you're going to have to know: diploid and haploid. Do y'all know how many chromosomes humans have?
23.
We have 46. We have 23. We have two sets of 23. Because we have two sets, we're diploid. Di means 2. Okay. Haploid is 1. Yes, sir.
So mitosis is just separation of the nucleus?
Yes.
Okay.
Yep. Now, cytokinesis, that's the division of the cytoplasm, resulting in two identical daughter cells. When we get into mitosis, we'll learn a little bit more about that in depth. Okay, so again, this is mitosis and cytokinesis. You see DNA replication, chromosomes align, they go to opposite ends of the cell. You have the division of the cytoplasm and the division of the nucleus. Remember, mitosis, division of the nucleus, cytoplasm, division of the cell. Okay. All right, recap and summary. Cell division is part of the life cycle of virtually all cells. Cell division is a process in which one cell divides to form two new cells. There are two main types of cells, prokaryotic and eukaryotic, each with its own process of cell division. What do we call it in prokaryotes? Binary fission. What do we call it in eukaryotes? Yeah, mitosis and cytokinesis. Okay. Yes, sir.
Isn't meiosis like the opposite of mitosis?
Not necessarily the opposite. So mitosis makes genetically identical cells that are both diploid. So the original cell for mitosis has two sets of chromosomes. The daughter cells in mitosis have two sets of chromosomes. With meiosis, that's a specialized type of cell division for gametes. You know what I mean when I say gametes? Spermate. Okay, so essentially what happens is they go from diploid, having two copies, to having one copy, haploid. The reason that's important is because that is how fertilization happens. You get half of the genetic material from one parent, half from another. So the process of meiosis does two different things. One, it splits the amount of genetic material, so there's just one version of each chromosome. Instead of having two sets, you now have one, so that when it comes to fertilization, the new fertilized egg has those two sets, half from mom, half from dad. The second reason is it causes, there's certain steps in there that cause genetic variation, which is extremely important, especially when we get to the genetics piece or the evolution piece, because what would happen if we were all exactly the same?
There would be no like.
One, somebody said, so that'd be super boring. The second thing is let's say,
Incest.
no, well, I'm not going to say you're wrong. I wouldn't use that word, but there's such a thing. You have issues when you have narrowing of the genetic pool. But also, let's say there was a disease that killed off people that had brown eyes, and if everybody was genetically identical and everybody had brown eyes, and what would happen to that disease? Yeah, extinction. Versus if we have people that had blue eyes, brown eyes, hazel eyes, or whatnot, they'd be able to survive and then propagate and so on and so forth, right?
I've never seen purple eyes.
Prokaryotes divide by binary fission, which includes DNA replication, chromosome segregation, and separation into two daughter cells. Eukaryotes, mitosis and cytokinesis, ensuring the daughter cells have all the necessary chromosomes and organelles to survive. All right, any questions on that?
It'd
Okay.
be like a whole day for your class.
We usually get to section and a half a day. Okay, on average, right? All right, the cell cycle. This is not a bicycle made of cells. All right, we're going to learn the phases of the cell cycle as well as how it is regulated.
So it's unicycle made.
Yeah, it's a unicycle made out of cells. You got it, right? Okay, so it is a repeating series of events, including growth, DNA synthesis, and cell division. This figure here details the cell cycle. We've got G phase, or G1, S phase, G2, and this is called the mitotic phase, cell division, right? Now, one thing about this is G phase, S phase, and G2 collectively Scientifically, they're called interphase. So G1, S, and G2 is called interphase. This is where normal cell functions happen. If you have a liver cell, this is where it breaks down, this is where it releases hormones. If you have a heart cell, this is where it's doing the contraction of the heart, right? Normal cell function. Now, granted, a big piece of this is DNA replication, which you need to get ready to divide.
So,
Because if you don't have your DNA replicated, what happens after this?
nothing.
Yeah, well, your cells will divide and they won't have enough genetic material to function, right? If you miss genetic material, you're not going to function very well. Yes, sir?
So, G1 like these are kind of the same thing.
Pretty much. So G1 is probably a little bit more of your normal functions, but it's going to be cell growth and getting bigger, right? G2 is you're specifically preparing for this, but you're still doing the same cell functions. Okay. Does that provide a little bit of clarity? All right, eukaryotic cell cycle subdivisions. All right, mitotic phase is M. Includes mitosis and cytokinesis. This is where the cell divides. Again, mitosis is the division of what? The nucleus. Cytokinesis is the division of the cytoplasm of the cell. All right, interphase. This includes GAP1, also called G1, synthesis S phase, and GAP2. This is the phase for growth, routine life processes, and preparation for cell division. So at any one time, most of your cells are in interphase, either GAP1, GAP2, or S phase. This is also the longest phase of the cycle.
Same purposes.
Yeah.
Or more.
Now, there's something, as an AP student, you probably need to know, there's a phase within interphase called G0. That is a specialized phase for neurons. Neurons are going to be the thing that make up your brain, your spinal cord, basically connect information from all parts of your body to the central processing unit, which is your head. It would be up here. What this blue line is or whatnot. These, or actually probably be up here. These will not divide. G0 is one that's like, all right, we're staying here, we're not planning on dividing, we're not planning on growing. This is where we're at. We're in G0 for the rest of our lives. Because the thing is, it is unique about neurons, and why like brain injuries or spinal cord injuries don't heal, neurons really aren't made to be replicated. Like once you got them, they're there. So however many brain cells you got, ladies and gentlemen, that's it. It's only going downhill from there, right? So
concussion is less.
yeah, absolutely. Here's the thing: your brain stopped forming in your mid-20s. Okay, so once you hit your mid-20s, your brain power, that's it, buddy. So something to think about as you're getting older. The more academically and more stimulating you do stuff with your brain as you get to 25, strengthens it, helps it kind of differentiate, helps it get a little bit better. And then once that happens, it's like, this is what I got. And it's a little bit harder to meld with and mesh with at that time. Like your brain, as you're getting older and developing, it's what's called fluid. It's like, yeah, you can, that's why it's so much easier to learn new languages when you're younger, because your brain's still growing and forming new connection. Once you're older though, it doesn't change as much. You don't make new neurons. You can change a little bit, you can make some new pathways. But for example, if you learn a language as you're getting older, there's a part in your brain that is specifically for that. For example, if you have a person that's bilingual and they learn both languages as they get older, that part of the brain, like, all right, all of this is both languages. But if you learn it as you're older, your brain has to be like, all right, I'm squishing English here and I'm going to allocate this to Spanish, right? And so it's not quite as efficient. You can still do stuff, but like your peak mental capacity, 25. You go down from there. Okay. Doesn't mean older people are not as smart, but like in generally their IQs drop, but they have what are called like, they have basically experienced intelligence. And it's like, oh, I did this once 30 years ago, I can use that to solve this problem that I'm doing today. Okay. So that's kind of the way to say what?
Wisdom.
Yeah, we'll say that. Right.
Moral of the story, don't be sipping shots.
Alright, so mitosis, the nucleus of the cell divides into two daughter nuclei. Cytokinesis, again, it's a split in the cell. So this here, mitosis is complete. You've got two different nuclei. And in the next one, it is cytokinesis. The cytoplasm splits into two. Alright, stages of interphase. GAP1, rapid cell growth, routine metabolic processes, preparation for DNA replication. What phase does DNA replication happen in?
G.
S. It does happen in interphase, but there's subdivisions of interphase.
How many is
Okay.
not wrong?
Yeah, synthesis is your S phase. This is where DNA is copied by DNA replication. This is a process occurring before cell division in which DNA is copied.
It's very important, I guess, if we don't enforce DNA's copy.
Yep, DNA is copied. Alright, so G1 phase, rapid growth, S phase, DNA replication. Gap 1, cell grows, proteins are made, routine metabolic processes. This is where your cells usually are. S phase, DNA is replicated. So as you go from gap 1 to gap 2 synthesis, we replicate the DNA. Gap 2, your final preparations for cell division are made. You make sure you organize your organelles, make sure you have 2 sets of them, etc. So you can go into mitosis. Okay. Any questions on that? Alright, remember, yes sir.
When does this happen?
All the time. So essentially, every single cell in your body is within one of these phases. It's either in G1, G2, S phase, or mitosis. Mitosis is the splitting of the cell, so it's just performing normal functions. It's either G1, S, or G2. All those together are called interphase. So like if you ever look at most of your cells, like if we look at this original picture up, oh no, not here. Never mind. This is the last section. If you look at a picture of a bunch of cells, most of them are going to be interphase, which is either G1, G2, or S phase. All right, again, gap 1, gap 2, G1, G2, and S phase are all part of interphase. And then you've got that mitotic phase where the cell actually splits.
Well,
All right, so this is kind of a little gift. So it's in G1, just hanging out. In S phase, it is starting to duplicate the DNA. G2, it starts to prepare for mitosis. Mitosis, the cell splits. Okay, and you're back to G1.
that thing got me.
Alright, so control the cell cycle. This is done by regulatory proteins. They control the cell cycle by signaling a start or delay of phases. Basically, hey, we're at G1, can we go into S phase? Or we're at S phase, can we go into G2? This ensures the completion of a phase before moving on to the next. Okay, there's going to be various checkpoints, including cell growth checkpoint at G1, DNA synthesis checkpoint at S phase, and the mitosis checkpoint right before mitosis. What do you think happens if these checkpoints break down?
Not bone, but.
Cell division stops.
What doesn't happen right?
Cancer. Okay? It does the opposite. Cell division keeps going. It's called uncontrolled cell division at that point. That's how you get cancer. Okay? So again, we've got a cell growth checkpoint at the end of gap 1, a DNA synthesis checkpoint at the end of gap 2, and then we've got your mitosis checkpoint during mitosis. Gap 0, this is the gap phase that neurons are going to be in, because once they're there, they're there. They're not going to really grow, they're not really going to divide, etc. Okay? Alright, cancer is a cell in which diseases grow out of control, usually because of mutations in genes that control the cell cycle. Yes, sir. A piece of this. Yes, sir.
And what's the chemo supposed to do this?
Okay, can we wait till after we get done the session, we'll talk about it? Okay. Causes include what causes an unregulated cell cycle due to DNA damage. The effects here will be the rapid division of cancerous cells, which form tumors, which are abnormal masses of cells that may be cancerous. Basically, this causes damage to tissues and organs. The way it does that is it's kind of like if you have a brain tumor, your brain's supposed to operate, but you have a bunch of cells growing in there. What's it gonna do to that brain? It's gonna start pushing on it, start to cause it to break down, break apart, etc. Right? So essentially, tumors just grow so fast and they muscle out and they push out all these other cells. They like clog them up or something. All right, recap. The cell cycle is a repeating series of events. It goes through. It includes DNA synthesis and cell division. In eukaryotic cells, there are 2 growth phases, gap 1 and gap 2, and cell division includes mitosis and cytokinesis. It's controlled by regulatory proteins at key checkpoints, gap 1, S, and mitosis. The proteins signal to either start or delay the next phase of the cell cycle. Basically, if it says, hey, do we have enough DNA? Yes. All right, cool. We can move on to the next step. Hey, are we big enough to start dividing? Yes. Great. Proceed on to the next step. But if the answer is no, we stop. Cancer is a disease that occurs rapidly when the cell cycle is no longer regulated. Cancer cells grow rapidly and may form to a mass of abnormal cells called a tumor. All right, to answer your question, so chemo is basically a poison for cells that specifically targets rapidly growing cells. The reason that it is used is because cancer cells go through what's called unregulated growth. They just keep on multiplying, keep on multiplying. So the drugs and chemo are targeted for those cells that grow the fastest, which are usually going to be the cancer cells. Right? Now, it poisons every single other cell of your body. Okay? But since they're not as rapidly growing as cancer cells, they're not as heavily as effective. Okay? Now, the thing is, what do you lose when you go through chemo? Your hair. Why do you think that is? Your hair growth is rapid compared to the rest of the cells in your body, so your chemo kills those cells that grow hair. Okay? Because it's targeting cells that are growing rapidly. Yes, sir.
So if we were to delay or stop cellular division, would that just make us not immortal, but aging?
I'm gonna say no, because listen, if you stop cell division, you're gonna die. Because here's the thing, pretty much every single cell in your body gets replaced, with the exception of neurons and some heart cells, gets replaced every 7 years. So your skin cells get replaced about every 2 weeks. And the reason is once they get damaged, it's not like they repair themselves. It's like, all right, we're just gonna get new cells here. Okay?
Is that why they say your taste buds change every 7 years?
Probably. I'm guessing. I don't know for sure.
Sorry, I was saying what?
Wait, so what's
So much
the
for
radio?
immunity.
The other one. So that's why.
Radiation?
Radiation.
So that is essentially where if you can isolate a tumor, then you can irradiate it in a specific area. So let's say this entire table is your body, your tumor is here. You get to irradiate just where the tumor is to kill those cells here. Okay, it's basically targeted killing of cells. Now, there aren't as many side effects with radiation that I know of, but granted, I'm not a cancer expert. But they're able to target it in one area. Because if you've got cancer throughout your entire body, they can't do radiation.
Yeah.
It's got to be within one area. Like, all right, we're going to radiate this tumor at the spot where we're having cancer. Okay. Does that answer that question?
Yeah.
So.
Yes, sir.
The thing about it being every 7 years is not true.
What?
Every 2 weeks.
Oh, wow. That's
For
weird.
what?
Yeah.
They what?
Every 2 weeks they change?
Yeah.
That's why we do like Chinese 1 week and then
Yeah.
Italian the next or something.
Some neurological stuff there. But depending on the part of your body, your cells get replaced at different rates. For example, like your muscle cells are in general slower. Your skin cells get replaced really quickly. Your stomach cells get replaced very rapidly because what kind of environment is in your stomach?
Very
Acidic.
Very acidic, right? So those of having to get replaced are located very often. So. All right. So you got those review questions for what sections were they?
3.31 and
3.1 and 3.32. Again, I will. How do you want me to do this? Just post a Google Doc?
Yeah.
Okay.
Which ones they are?
No, I'm not gonna do that. Like, yeah, I mean, you're, because I don't know how far we'll get in a week. Does that make sense?
Like when you sign the secret, you have to do.
Yeah, I'm about to say.
Basically.
That's pretty much most of it, right? We probably won't have class tomorrow.
Yeah. I get.
Yeah, so create. We've got assembly.
Here, why don't you grab the stuff for it?
I'm just going to call it week 13 review questions.
Yeah, why don't you grab it?
I know you said that you can't, like, give me any sort of medication, but if I were to call my grandma.
I don't know what it is. I think we have to have paperwork filled out.
What? So she can't come in here and explicitly say, like, in front of a camera and everything, I need all sorts of medicine.
I know. I made a mistake.
Yeah, technically everybody does here.
See ya.
Very important case.
Are your cells divided into two close?
Yes. I wish I could draw a flower.
I don't know, but I'm gonna eat some chicken fried rice. Yeah, give it to me.
You telling me a chicken fried that rice?
Huh?
You telling me a chicken fried that rice?
Yeah.
See ya.
See ya.
See you later, Mr. Rowell.
Mhm.